of the genome and its regulation in different biological concepts. I would like to introduce our session chairs. I would like to introduce our session chairs, Professor Arup Bandopadhyay and Professor Radha Chaudhary. I would now like to introduce them with a brief introduction. Professor Arup Bandopadhyay was former head of the Department of Anthropology, University of Calcutta. He served as president of the Anthropological and Behavioral Sciences section at the Indian Science Congress's Senatory year. His research spanned evolutionary biology, forensic and dental anthropology, medical genetics, cytogenetics, dermatoglyphics, public health, population genetics, and human rights. Professor Radha Chaudhary is from Department of Zoology, Institute of Science, Banaras Hindu University. Her expertise lies on molecular reproductive neuroendocrinology, behavioral neuroendocrinology, enzymology, reproductive endocrinology, and endocrine disruption. I request both of you to please have your seats on the dais. To set the tone for this session, we are honored to welcome Professor Shrikant Mane, Director of Yale Center for Genome Analysis, YCGA, and Tech Proteomics Laboratory at Yale University, with a PhD in cancer biology, 1985, and a postdoc from Johns Hopkins. Dr. Mane has over 25 years of experience in genomic and proteomic research. He founded YCGA in 2009, leading it to global recognition. I invite you, sir, to please come on stage and share your insights on increasing diagnostic rate in rare diseases, lessons learned, and defining variants of unknown significance. Professor Mane, the stage is all yours.
Thank you for the kind introduction. I also wanted to thank you, the organization, for giving me this opportunity. So far, I'm having a lot of fun and I learned a lot of things. I would like to particularly thank Dr. Nyan. So, without taking too much of time, I would like to start with my presentation. I'm going to talk about most of the research has been focused on rare diseases, and recently I moved to cancer genomics. So in 2009, when Obama became a president in 2008, I was able to attract hundreds of millions of dollars through stimulus funding, as well as that time I had published exome sequencing technology. So because of that, the Yale University asked me to establish the Genome Center, which I did that in 2010. And I just quickly want to go through the Genome Center, because the Genome Center is key to most of the work which I'll be talking today. So as you can see that we have very great laboratory. This is the inset from the class. We have all the cutting edge NGS platform, including Illumina, PacBio. We also have single cell genomics and spatial multiomics. We also have dedicated computer infrastructure, because without that, you can't do any genomics work. So we were the first CLIA certified lab in 2011 for NGS applications. And currently we are generating almost 1 petabyte of sequence data each year. The use of the facility by Yale as well as other researchers from over 80 countries have resulted into more than 2,000 articles. Many of them are high-profile discoveries which are appeared in journals like Nature, Science, Cell, Human Development, Nature Genetics. And most importantly, as usual, USA is a very capitalist country, where everything is counted in terms of money. So really, YCG has almost attracted more than $1.5 billion extra funding. I myself have attracted almost close to $100 million. So So YCGA, the Genome Center, has been really in forefront of identifying disease-causing mutations, but it also is in the forefront of starting genomic era. So we have developed many protocols, first time like FFP, you must have heard the dual indexing, index hopping, all those terminologies were determined by us. But my real work started in 2005 when I published this paper. It was on the cover of Science, and which identified the genetic SNP which was associated with age-related macular degeneration. So this was the first publication after the completion of Human Genome Project, which actually gets the credit for starting current genomic and precision medicine era. So in 2009, since working in a medical school, that time the whole genome sequencing was still like $5 to $10 billion. It was very expensive, not to mention nobody had infrastructure to manage that data. So I argued with a couple of researchers at Yale that why sequence the entire genome? Why not just focus on the exome, which has almost 95% of disease-causing mutation? So we worked collaboratively with Roche, that time it was Nimble, and developed exome sequencing technology. This paper came exactly at the same time as Jay Shenduri's paper, and although our publication has a lot more technical details than Jay's, by the way, Jay Shendu is also from India, and he happens to be from the same town I hail from Maharashtra. So it wasn't too much of coincidence. Both of us published the same paper within 15 days of each other. So this was also the first publication where exome sequencing was used for clinical application. This was the actual example where we took the clinical sample and proved the diagnosis made by the doctors was wrong and accurately diagnosed the patient. So I want to switch the gears now. Because of this genome center and the exceptional facility and exceptional scientists at Yale, uh, I was able to attract a significant amount of funding from National Institute of Human Genome. The total funding was approximately $25 million, and it was two rounds of funding. And the reason I want to tell you because this actually grant allowed me to collaborate with many of Indian scientists because this grant specifically sequenced the sample free of charge and no strings were attached, data belonged to the investigator. So we sequenced almost 100,000 exomes through this program, through this program, through this program and over 500 phenotypes. And after two rounds of funding, NHGRI decided that why keep sequencing samples when we are not able to identify the disease-causing mutation in more than 50% of the cases. That's why they decided to switch to the functional, high-throughput functional assay. And this is the topic of my today's talk. What is so important and what is the challenge at hand? Just sequencing really doesn't help. So basically, so most of the rare diseases, these are classical genetic diseases. There's 100% genetic cause associated with these diseases, rare diseases, because that runs into the family. Nonetheless, despite that, most of the cases, most of the diseases, we were able to identify variant associated with disease only 20 to 50% of the cases, depending upon the phenotype. Some phenotypes were actually very easy. And I think this is really a challenge because this really indicates the limitations of this exome sequencing or sequencing approach at that time. It looks like I lost the voice, but I hope you can hear me. So basically, if we want to realize the importance of the precision medicine, there is nothing like rare diseases because these are 100% genetic diseases. If we can identify the disease-causing mutation in more than 50% of the cases, there was no point in just keep sequencing the samples. So the third time when the NHGRI announced this Mendelian genomic RFA, they decided to specifically focus on developing high throughput functional assays rather than keep sequencing the samples. And so basically, so as I mentioned, exome sequencing is a great tool. It really offers you the really good view, allows you to integrate the coding region. But the problem is, as you know, the exome sequencing doesn't cover 100% of the exome, especially repetitive regions and other regions. And there's not uniformity of the coverage because there is a PCR involved, so doing structural variation is also a challenge. And most importantly, non-coding region is not accessible by exome sequencing. So these are the limitations of the exome sequencing. So then what could be the reason? Where is this missing heritability or the genetic markers which are causing the disease? Where they could be hiding if we can't find in exome data. So what we have come up with the strategy is that we periodically, every 2 or 3 years, we reanalyze our whole data with the latest, and I'll be talking about each of these slides in future. We're also looking at the whole genome sequencing to look at non-coding region as well as getting the complete coverage of the exome also. We're studying structural variation. We are also looking at the methylation changes, and we are doing also RNA-seq. Today I won't have time because I have only 25 minutes of time. So other issue is that even Mendelian inheritance is not as simple as it was described by Mendelian. It's very complex because there are a lot of complex mode of inheritance, variable expressivity, trait caused by de novo as well as somatic mutations. So that complicates the matter. And in the end, many of us already know what is variants of uncertain significance. These are the variants which are present in the exome as well as whole genome data, but we cannot prioritize or we cannot determine whether they are causing the disease because we don't know the functional significance of those variants. And that's what is the key challenge at the current moment. So periodic reanalysis, as I mentioned, we routinely reanalyze our data. And I think because one of the reasons is that because analytical pipelines are updated, there is a lot of information available in literature, new tools are being developed. So we utilize this tool to see whether we can really identify the variants which are not identified in the exome data. For example, in this case, we have the polycystic liver disease cases, 128 WS negative cases, that means where we're not able to identify the variant using exome sequencing. We reanalyze using this exome hidden marker model, and we are able to identify 25 kb heterozygous novel pathogenic deletion in GANAB gene. So the important thing again, many of our publications are in Nature, Science, Cell, or New England Journal of Medicine, is not just by identifying the variant, but we go beyond that and try to prove by doing functional assays that variant is actually causing that disease. So interval of greater than 2 years is ideal because in 2 years not much happens. 2 years there is enough literature available or the new pipelines are available, so we can reanalyze the data. So we also have looked into doing whole genome sequencing for many of thousands of our exome negative cases. And as you know, the whole genome sequencing, especially PCR-free, is really good. It offers a lot better coverage for even coding region. It offers information about non-coding region. It also allows us to detect structural variations better than the exome sequencing. So I want to show you one particular case. This is from Joe Gleason's lab. He was also my collaborator in Mendelian Genome Centers. Two siblings from a consanguineous family with microcephaly cerebral hypoplasia were unsolved by WS analysis, so we decided to do the whole genome sequencing. And as you can see here, that these are the two siblings. You can see there is a 14.2 kb deletion here, and this is parental DNA. So the deletion is absent in parent, is only in the sibling. And this deletion is actually in WSB2 gene, which is a ubiquitation WDR family gene, which definitely responsible for neurodevelopmental aspect. And when we really did the RNA expression analysis, obviously this deletion completely knocked off the expression of this particular gene. So obviously it was very easy, and I think they have done the further functional study to show that this deletion by doing CRISPR that does cause this particular disease in model organisms. So again, even though exome is better than whole genome, it still has many limitations because it's a short read sequencing technology. Structural variation identification is not that great. And I think it's not good for the repetitive reasons such as segmental duplication, tandem repeats. And these regions are very important clinically because there are almost 1,000 genes present in this. Again, as I mentioned, not ideal for structural variation. Relevance of non-coding variants. Even if you identify the variant which is in non-coding region, we still don't know what is its functional aspect. So that's why again I'm coming to the last point, and which is the main point of my today's talk. Um, it doesn't really improve the solve rate significantly because the issue is variants of uncertain significance, because there are so many variants in this data, both exome as well as genome, we don't know. We can't, bioinformatically, we cannot prioritize them to be a causal variant. So then optical genome mapping, you must have heard the bio nanotechnology. That has been also used to identify variants associated with so there were two cases unsolved by microarray exome as well as whole genome sequencing. So this is where they use optical genome mapping technology. I don't know whether you know, they really introduce fluorescent dyes at fixed interval and they scan it. And as you can see that they were able to identify inversion here as well as the deletion in this particular gene, which is again responsible for neurodevelopmental diseases. But the optical genome mapping has many limitations. First of all, it requires very high molecular weight DNA. It's very difficult to get when you have small biopsy. It's very efficient for identifying mosaic structural variation, which are difficult to detect both by microarray as well as sequencing. But the resolution is limited because the fluorescent dyes are actually, they're too far apart, so you can't screen the entire area. High molecular weight DNA, and the most important thing is that this is just the first step to identify, because ultimately it doesn't give any idea about the structural variation. You still need to do the sequencing. So this is good as just the original scan. So again, long read, I think you must have heard this debate whether to do long read sequencing or short read sequencing. Both technologies has pros and cons. Two popular platforms for long read sequencing, PacBio and Oxford Nanopore. They are single molecule sequencing technologies. So basically the advantage of single molecule sequencing is you don't know PCR, so you don't get PCR biased. And most important thing is that it's very good at identifying structural variation because you are getting very long stages of DNA. It also can do entire transcriptome if you want to do alternate splicing. And most important thing, while you are doing sequencing, because it's a single molecular sequencing technology, you can also read DNA modifications such as methylation. So overall, it's really good technology. And these are the two examples I have shown here. They were able to identify hemizygous inversion here on X chromosome and neuromuscular disorder. Also, in other case, they identified a duplication here. These kind of changes are very easy to identify by long read sequencing. So, but again, there are many limitations. First of all, you require large amount of DNA, very high molecular weight DNA. Then again, the error rates are still high, even in PacBio, with CCS reading, the error rates are significantly low, but if you take at the raw consensus, error rates are still high. It's extremely expensive. Illumina whole genome in my lab costs $300. With PacBio review, it costs something like $1,700 to $2,000. And the data analysis is still a challenge. It's not a routine like Illumina. So now I want to move to the next thing is like DNA modification, because there are many diseases, especially psychiatric diseases, as well as cancers are caused by changes in methylation pattern or DNA modification. So this is a very classical case studied by again Mendelian Genome Center, Debbie Nickerson from University of Washington. So they analyzed like 4 cases. They were Baratella spot syndrome. They were able to identify the variant. XYLT1 gene is very known to cause this disease. Then did whole genome analysis in 2 additional cases they were able to identify, but there were still 2 cases where they were not able to identify the disease-causing variant. So when they tried to amplify XYLT1 gene, they realized there was PCR amplification bias, as well as some SNPs were missing. At that time, they realized there is some structural defect or some DNA modification which is causing this. So they did the restriction enzyme for methylation, and they were able to identify the changes in the methylation. And to look into further, they did the whole genome methylation, and what they were able to identify that there was expansion of GCG repeat in the promoter region of this particular gene, which caused hypermethylation in exon 1. And I think that was the reason when we did RNA-seq cDNA, then you can see that the exon 1 was completely, expression was completely abolished, and the expression of the whole transcript was also significantly reduced. So that's why I think they were able to identify. And I think we have done this approach for many other neurodevelopmental diseases, especially. And we have identified the methylation pattern. But again, as I mentioned, identifying methylation pattern is much easier with PacBio than the Illumina system. So, the last thing actually, this is from Daniel MacArthur's lab. Now he's moved to Australia. He was also part of the Mendelian Genome Center. So, exome definitely identifies the exome as well as whole genome variant. But the problem here is that many intronic or deep intronic mutations, especially slight variants, actually you cannot figure out the functional role of that. So in this case, if you do the RNA sequencing, then it adds the functional information to the genomics data, and then it's easier to identify the mutation, and is again we are doing sort of functional analysis of the variants. So in this case, actually there are 15 exome negative cases and rare muscular disorder, and they did the RNA-seq because in the muscular diseases it's easy to obtain the affected tissue biopsy. So they did the RNA-seq, and they were able to identify 17 cases, were able to diagnose with RNA-seq. As you can see here, one of the example I have shown, this is a control, this is the standard alternate splicing. But in this case where the disease, you can see there is additional site here, C to T transition here, which actually resulting in a stronger splice motif that results in the introduction of a premature stop codon of the transcripts, and the transcript is obviously not expressed, and this causes the disease. So they were able to report almost 30% diagnosis rate above exome and whole genome using RNA-seq. But it's still challenging, and one of the limitation of this is the accessible tissue. If it is something skin or something superficial, you can access the affected tissue, but if it is something internal organ, it's hard. And of course, there are various ways you can study, like iPS cells or fibroblasts or circulating blood cells, lymphocyte, but not all genes are expressed in these tissues. So it still has a limitations for this technique. So despite using multiple approach, I already told you we did whole exome, whole genome, methylation analysis, long read sequencing, bio-nano, or even RNA-seq. RNA-seq probably certain diseases, actually center condition is very helpful, but most of the condition is not. So the solve rate is not increased significantly, maybe 10 to 20%, not much. So basically we come back to the same thing, that there are still 40 to 50% of the cases where we are not able to identify the cause of the genetic disease. And I think that is why I think functional characterization of the variant is very extremely important. So as you know that we all do the functional analysis. There are so many in vitro studies as well as model organisms, as well as we have electrophysiological approaches, as well as you can do organoids. But these are very low throughput approaches. So when you want to study something like this, for every variant there has to be specific study design, and that limits the high throughput approach because there are so many variants of uncertain significance, hundreds and thousands of them depending upon which data set you are looking. This is not the approach you need to take because it takes years and years to study even one variant. So this is why I think the important thing is there is a need. There is a really pressing need to develop high throughput functional assays. And this is, I just wanted to give you some example that how little we know about the variants. So for example, 8 billion people carries almost 9 trillion possible single nucleotide variants. So far, only 1% of that human genes or the variants have been functionally characterized. So we still have 99% of the work done. Okay, so that's why, as I mentioned, NHGRI is focusing more on developing functional assays. So there is a need. So the new functional assays have been developed now. This is Doug Fowler and other people from Stanford as well as Europe. They have come up with this term called multiplex assays for variant effects, called MAVs, such as you are already aware of this deep mutational scanning, massively parallel reporter assays, and saturation genome editing assays. These are the type of the assays which have been used. Again, this is a very classical work done by Jay Shenduri about BRCA1 characterization of BRCA1 variant of uncertain significance. So what is the advantage of these assays? So first of all, they are highly scalable. Variants are synthesized and analyzed.
Dr. Mane, you have just 2 minutes more. Can you finish?
So I think I had 25 minutes, right? And I'm looking at the watch.
20 minutes I have been.
25 minutes. That's
25 is all right?
Yeah. So 25 minutes is what I was told.
Okay.
But I can finish it if you want.
Okay, okay. I don't know. I have
No,
to be informed as well.
I know. That's nice of you, but I'm looking at the watch too. So basically, these are highly scalable. You can synthesize large-scale variants in pool format and 9612 format. They are high throughput. Thousands of variants can be screened in one assay, and most of the assays use NGS readout, so it's very easy to do it. And these assays are becoming very popular now. And there is an alliance firm. It's called Atlas of Variant Effect Alliance. Because precision medicine has to be effective, this has to be done. So the vision of the alliance is to test nearly all possible single nucleotide or amino acid variants. I'm not talking about what is known, but I'm talking about all of them, and that's possible only using maps. Okay, and this brings together a lot of different type of scientists, computer scientists, biologists, and everybody else. I don't know why there is something. So I think NHGRI as well as other European organizations are paying really a lot of attention. There are Gregor consortium as well as IGVF consortium. NHGRI alone has spent $185 million to fund these projects where they are developing this kind of high throughput assays and trying to analyze the structure. Okay, and in addition to this, so there's no point in having, if there is no some kind of registry or data repository for all this information. They have been also developed. So basically, the infrastructure for doing this kind of assays and sharing the data with the entire scientific world is actually already in place. Okay. So I just wanted to summarize, and I guess that will end my talk on time. So again, we can go beyond exome analysis, do whole genome sequencing. Many of my Indian colleagues, they are very, very fond of doing whole genome sequencing. Probably that's a fancy word, but I have done the whole genome sequencing for at least 50 cases. None of them have been able to solve the case by doing whole genome. Again, use the analytical tool. That definitely helps. Look for new model of inheritance. We have done for skin as well as other congenital heart disease. Actually, this is a big project I have in collaboration with India. We see a lot of epigenetic changes, de novo mutations, different mode of inheritance. That has definitely helped us. But there are major limitations despite that. Because the major limitation is that, again, this is the classical case that, okay, we know we sequenced the genome long time ago, but we still don't know that almost 5,000 to 10,000 novel genes for Mendelian conditions we still don't know, because Mendelian conditions are very polygenic. There are very heterogeneous conditions. They are caused by defect in one gene, but there are many genes which cause the same effect. There are 6,000 to 40,000 novel Mendelian conditions still remains to be identified. So that is definitely causing a problem. Analytical tool is still an issue because prioritization is a challenge, but that's mostly because of lack of functional information. And reliable high throughput assay for characterizing the functional role of variants of uncertain significance. And the most important thing, I know that I think this is happening now better in India, they are sharing data. But unless you share the data openly, because any NIH grant now you get, you are forced to share the data within 6 months of completing the work. No excuses, otherwise they cut the grant funding. Because once you share that you identified a variant for a particular, say for example, nephrotic syndrome, then rest of the world actually really knows that this also variant can cause the disease and they can add to their list of the gene to be screened. So again, I want to thank you, organizers, for giving me this opportunity. My collaborations with Centers for Mendelian Genomics, my staff as well as university, and I also want to thank all the grant funding I have received from NIH. So if there are any questions, I will be more than happy to answer them.
Thank you, Dr. Mane. It's mind-blowing and a rare presentation because it's a rare disease. So I can take two rare questions, right? Excellent and rare questions.
Thank you, Shrikant. That was a wonderful talk. Yeah, I sense calling variants has been a kind of a major concern. So whilst we could do or call variants from RNA-seq data, the variants that are called from RNA-seq are probably not that tangible when compared to say whole exome or whole genome. So do you see a challenge between the low-pass sequencing with a depth of coverage of say 1.5x or 2x, and a mixture of a targeted genome approach taking the RNA-seq, and then taking or looking into those particular variants and prioritizing them again later in the book. So where do you see the low-pass sequencing integrated into this, so that at the end of the day, we are looking only at the bona fidelity of those particular variants that are called, so that we're not only looking beyond the VOS, but we're also going for not expecting the conflicting, interpreting variants, conflicting.
That's a very interesting point. I think nowadays we are already doing low pass, but mostly we are doing that with long read sequencing because for structural variation. Because Illumina is pretty good with identifying variants.
And yet again, I'm sorry.
I know this is
even the states, couldn't publish this particular data in the public domain yet. Not many did.
So when you talk about clinical thing, it's very rigid because when we do research exam, we do at 30 to 40x. So when it is clinical, it has to be minimum 60x. Especially the coverage for the clinically relevant gene has to be almost 200 to 500x. So this is one of the reasons, because if it is clinical, forget about it. You have to have that kind of redundancy. And one of the reason is that, as you know, especially for somatic variants, you need to have very high coverage. And nowadays, actually, earlier we used to use confirm our NGS data with Sanger sequencing. Not anymore, because we found that NGS data is way more.
I'm sorry, I'll just take one more. How much would
I think Prakash, first we just go for
I'll be
another
happy
one
to
and
talk
just another one to close this one.
CPG islands, right sides of the exome, mostly 90% of them are there in the promoter regions. That slice is an AG and GT. An interesting observation, can you explain the AG and GT, the C system? So there's no possibility of CPG utilization in the slice sites. The slice is the AG and GT.
I can't hear you because there's no mic is not okay.
What you need to say is the splice sites are AG and GT. The splice sites, AG and GT. One species C is missing in the splice sites. If C were there, it would have been subject to CPG utilization. So, what is
Yeah, so exome sequencing also, there are different kits available, right? There are some exome sequencing kits really cover promoter regions, also regulatory regions. But the point of the matter, as I mentioned, just by identifying the mutation in promoter region are possible. If you don't know that that's a possible splice site, then it's very difficult to interpret. And that's why you need to do RNA-seq.
CPG islands indicate the beginning of coding region and promoter region. So, if there's no possibility of CPG utilization in the splice sites, AG and GT. So
yeah, but most of the, unfortunately, most of the exome really don't go beyond in that region. That's what.
To avoid the possibility of coding that the splice sites do not have a C.
No, I think it's all depending upon the cost and the relevance. So, as I mentioned, there used to be time when we used to include those regions also in the exome, but I think nowadays that's not included, especially for clinical exome.
Hello. Can we stop here for this? And Dr. Mane, I'm very much sorry because whenever I sit down here and speak in the audience, it is very difficult to sit down here for cutting the time.